All right, everyone, we're going to go ahead and get started. If you can find your seats, that would be great. I've got a couple more folks coming in the door. Um, but I just want to thank everybody for being here. My name is Cinnamon Moffat. I'm the Associate Director of Marine Research Support here at Hatfield, located in Newport, Oregon. This is a hybrid event, so we have folks online and folks in the room. And this is our first seminar of the academic season. So welcome back if you're my regular. And if this is a new event for you, we're so glad to have you. A couple of logistical details as we're getting kind of settled. For folks that are online, if you have any questions or any technical difficulties, if you put those information or those questions into the chat box, we have a volunteer in the room, Roseanne. Thank you back, Roseanne. She will help navigate those. And if you have questions for today's speaker, if you put those into the chat box, we'll make sure to get those read out so we can all hear them. For folks in the room, most of you remember this, but if you have a question, you're going to need to raise your hand and I will run you a mic, and that way folks online can hear. We do have students taking this session for credit. And so if you are a student either in the room or online, make sure that you either sign in on the form as you're walking in the door so that our instructor, Kate Stafford, can make sure that you are present. So that is a way to make sure you get credit for being here today. And again, just thank you so much for being here. Typically, I make some announcements, and this announcement I'm going to make is that we're not having a seminar next week, which is kind of sad. But after that, we were going to have a seminar every single week, same place, same time, every Thursday afternoon. So I hope that this becomes a regular part of your Thursdays. But again, we do not have a speaker for next week. But thanks for being here. Okay, I'm going to jump us right in because Chris has a whole slide deck that looks amazing and wonderful. And so I just want to do a quick introduction of our speaker today. I think most of you all know this is Chris Langdon. And Chris Langdon was born in Somerset, England, and then did his graduate degrees at the University of Wales before moving to the University of Delaware for a postdoc. And then soon after joined OSU in 1985 as an assistant professor with what was then named the Department of Fisheries and Wildlife, and was based right from the beginning here at HMSC. In 1996, he founded the Molluscan Broodstock Program to enhance yields of Pacific oysters on the West Coast. And I think out of everything, he's most known for, you know what, this Pacific dulls, which tastes like bacon when it's deep fry. And so that is definitely a tagline that is always associated with Chris Langan. And so without further ado, I'm going to hand this off to Chris. Welcome.
Thank you, Cinnamon. So yes, I'm more famous for those 3 or 4 words than for 40 years of research. But there we are, you know. We live in a world of conspiracies. So I'm trying to go and condense, you know, 40 years of research into 45 minutes or so. So bear with me. I probably will skip some subjects that are close to your heart, like dulse, for example. And I'm going to focus just on oysters because that's been kind of my current, my major focus of my research over the years. So the title here is Taming the Pacific Oyster in the Wild West. And I'm going to try and include as many of my colleagues and friends and students in the talk as possible. But if I do miss some folks, please don't hold that against me. I've got some timing I have to keep through. So domestication or taming, it's really about learning how to farm the animal or plant. And it lies at the heart of human civilization. So without our ability to farm animals and plants, a lot of our civilizations, cities and others would never have occurred. And so I'm going to talk to you a little bit about taming the oyster using a little bit of AI. So I've been having a lot of fun with this. There you go. Tally ho. So the oyster, Pacific oyster, is really, really important as an economic species globally. And it's been cultivated all over the world. And as you can see, production, I'm going to get my clicker working here. Production has been increasing over the years, a lot of it from China. So what we do here at the Hatfield Center has some global implications. The industry is worth about $8 billion in terms of harvest value each year. The first step in any farming activity initially is to identify the species and stock to farm. And some of you may know the native oyster is our native species. And here on the West Coast, it was a really important food for Native Americans. And unfortunately, during the late 1800s, there was intensive harvesting of this species by coastal settlers. And the population pretty much collapsed. So you can see in Willapa Bay here, it dropped to almost zero levels by 1916, and took a little longer in Puget Sound to drop to those very low levels. And the early farmers found that it was a difficult species to cultivate because it was very susceptible to freezing during the winter. That's why they had to build all these dikes. And it was also very slow growing and didn't have a lot of meat. And so folks started to look for alternatives to the native oyster because people liked eating oysters and wanted to continue that activity. And two of the great pioneers on the West Coast who brought and started to evaluate the aquaculture potential of the Pacific oyster on the West Coast were Trevor Kincaid at the University of Washington, and in British Columbia, Dan Quayle. And both these folks did a lot of the early work that was responsible for creating the foundation of what we see as our industry today. So in the early 1900s, there was starting to ship large quantities of seed from Miyagi Prefecture in Japan once people realized it was a species that grew well on the West Coast. And here we see it in Tillamook Bay in 1932 pre-war days, a oyster processing plant, the first one that was built here in Oregon. And here's a picture here of some of these crates of culch that was brought over with attached baby oysters from Japan. Now, some of you may know that the oyster has actually a complicated life cycle. The oysters that you're familiar with in stores and restaurants really is just half of the life cycle. These are the adults that are benthic, but there's also a planktonic larval stage that lasts about 2 to 3 weeks. And once the larvae reach a certain stage, what the oyster researchers and culturists say, they larvae set, which means they settle out of suspension attached to culch. I'm going to quiz you at the end of this talk for these terms. And then they metamorphose and become spat, which are baby oysters. So three words there that are characteristic of what we know about what we describe the life cycle. And in the early days, apart from the shipments of oysters seed from Japan, there was a couple of bays, Daybar Bay and Willapa Bay, where water conditions were suitable for natural production of the oyster, natural life cycle of the oyster being complete. These were bays where the water was warm and there wasn't a lot of flushing. So the larvae were able to complete their larval development. And here on the left here, we see some pieces of culch with attached spat. Now, this is a really interesting record going back to 1942 that was carried out in Willapa Bay looking at the set that occurred each year. And you can see that there are gaps. There are periods of 3 or 4 years where there was no natural set. So this meant that the oyster industry and the oyster farmers who were dependent on natural set or shipments from Japan were in trouble because there wasn't any seed available. So this first period in the 70s was the impetus for development of hatcheries. The second impetus here was caused the development of methods to combat OA. So hatchery techniques. So back in the 60s, 50s and 60s, there weren't really a good understanding of how to rear oysters. And again, we have two pioneers here who really set the scene and helped us develop some of these techniques. Victor Lusinoff, who was in Milford on the East Coast in Connecticut, he was developing techniques for rearing the eastern oyster. And Peter Warren, which is worked at the MAF Center in Conway, North Wales, and he was actually one of my advisors for my PhD. And both these folks who really developed a lot of what we know and currently use for rearing oyster larvae, Pacific oyster larvae. When I came to the Hatfield Center in 1985, I was lucky to overlap with some key people here at the Hatfield Center who were taking this technology from Europe and from the East Coast and transferring it to the West Coast so that we could develop our own hatcheries here on the West Coast. Willie Breeze was my predecessor. Anya Robinson worked with me for quite some time. Jim Lannon was one of the first geneticists who worked on oysters. Bob Maloof was a PhD student at this time and later became director of Oregon Sea Grant for a period of time. So these folks were very instrumental in the early days. And so this research led to the establishment of 4 major oyster hatcheries on the West Coast. Hawaiian shellfish in Hawaii, 2 hatcheries in Dabob Bay, Pacific seafood and Taylor shellfish, and then a hatchery in Oregon called Whiskey Creek. And Whiskey Creek is a very important hatchery because it supplies most of the small oyster farmers on the West Coast and internationally too, because the other 3 hatcheries are integrated into large companies. So their primary sales are to their own farms and farmers. So Whiskey Creek is very important in meeting the needs of many, many small farmers. And it's important for another reason, and I'll explain that in a moment. And Sue Cudd, who's here in the audience, she was responsible for maintaining this hatchery and keeping it operational over these many years. So I mentioned another reason why Whiskey Creek Hatchery is important, and that's because we were in the forefront, or they were in the forefront of a battle against what is going to become increasingly important this century, which is ocean acidification. And this ocean acidification is caused on the West Coast by upwelling. So we get winds from the north here in summer that pulls this deep acidified water to the surface. The water is also hypoxic. So here we see a cross-section of the water showing this deep water being brought up to the surface. And back in about 2005 or so, we started here at the Hatfield Center as well as at Whiskey Creek seeing some problems rearing oyster larvae. And this became really acute. By 2008, most of the hatcheries had a severe reduction in in larval rearing. And the folks at the Whiskey Creek Hatchery, Alan Barton, Sue and Mark Weigert, were really instrumental in observing the fact that the problems with oyster larval rearing coincided with upwelling events. And this triggered in some people's heads a realization that it was probably related to acidified water. So in these early days, I wanted to take my hat off to Betsy Johnson, who was an Oregon senator at the time. She was very helpful in establishing some funding for us, which kind of kept us going in those early days in terms of research on this problem. But we later subsequently got a larger grant from NSF to look at this problem in greater detail. Burke Hales, George Wolbusser, and Matt Gray, who's a PhD student in my lab, got together. And we were very fortunate. We were able to tease apart all the carbonate chemistry elements and separate them and look at them individually. And we found that aragonite saturation state, which is the solubility of aragonite, which is made of the oyster shell, larval oyster shell, the solubility of that shell in seawater was the determining factor in affecting the proportion of normal developing oyster larvae. And so with this realization, the oyster hatchery, Whiskey Creek, and many other hatcheries around the world now are treating their water. So the water is brought into the hatchery. It goes through what we're calling the Berkulator. This was an instrument developed by Burke Hales. The aragonite saturation state is determined, and then the seawater is buffered before it goes into the larval cultures. So this was a tremendous improvement in our understanding of the effect of water chemistry. And it's really one of the prime examples where we can actually put an economic cost to ocean acidification. So algal diets. The hatcheries at the moment depend on algal diets for rearing larvae. And going back again in history, in 1937, Mary Park in Britain was the first person to identify an algal species, in this case Isochrysis galbana, as a food that supported the growth of oyster larvae. And that discovery was really instrumental in developing various or many different algal species that are currently being used for rearing oysters larvae, and also a whole set of technologies associated with rearing or growing the microalgae. And currently we have in our facility here these algal bioreactors, which are really a godsend. They provide us with really large quantities of very high quality algae. So this technology has really taken hold. If you look on what the oyster's eating in the wild, what the adult oyster is eating in the wild, it's actually much more complicated. And so if this is some DNA studies of the gut contents of oysters in Korea, and you can see between 40 and maybe 75% of the diet is made up of diatoms and dinoflagellates. But there's a lot of other material in the guts of these oysters. And so the diet is actually very complex in nature. We did some work, this is going way back into the late 1980s, with Roger Newell and Daniel Krieger. And we looked at the importance of cellulosic detritus in the nutrition of eastern oysters in Canary Marsh, Delaware. And in terms of nitrogen, the bacteria particularly make quite a significant contribution. About 27% of their metabolic requirements are met by ingestion of bacteria, either free bacteria or bacteria associated with detrital particles. So we can understand a little bit about what oysters eat and how to rear them, but we still do not have an artificial diet for rearing oysters. And this is a huge dilemma, because the cost of algae is very high. It ranges from about $50 up to somewhere up there, $400 per kilogram dry weight, depending on culture methods and the scale of culture, etc. And the interesting thing, to me anyway, is that with oysters, you can't substitute living algae with dead algae. So there's something intrinsically important in live algae to the nutrition of these animals. So a big chunk of my career has been focused on developing microparticulate artificial diets for oysters and other marine suspension feeders like fish larvae. And here are 3 graduate students who were really critical in this work: Uma Nal, Matt Hawkert, some of you may know him, and Michael Buchel. And the challenge we have with these microparticulate diets is that they have a very high surface area to volume ratio. And this means that leakage rates are very rapid. So it's very hard to encapsulate these low molecular weight water-soluble nutrients like vitamins or vitamins and amino acids. And then you have a problem of acceptability. So oysters are choosy feeders. They don't eat everything they capture. Digestibility. And finally, you get to the point of, well, what do you put inside the particle once it's acceptable, digestible, and retains nutrients? So these are some of the particles we've worked on over the years. These microgel and particles and cross-linked protein wall capsules and coacervate wall lipid beads. These are all great for delivering the macro molecules that are high molecular weight: proteins, carbohydrates, lipids. These 2 particle types, the lipid wall capsules and liposomes, are very useful for delivering water-soluble vitamins and amino acids and other water-soluble nutrients at a low molecular weight. And complex particles is you take these 2 particles here and put them in a microgel particle. So that's why they're called complex. And these work. So here we've got, on the left, some pictures of some lipid spray beads containing riboflavin. This is a fluorescent water-soluble nutrient. And then here's some Pacific oyster larvae that have been fed those particles. And you can see their guts are all glowing happy yellow. They've had their vitamin supplement for the day. So we can at least deliver nutrients to these animals. It's just a question of, can we actually develop a defined artificial diet? And the answer is no at the moment. So a simpler approach that I've worked on but for my PhD was to take an algal species like Dunaliella that is deficient in a fatty acid, in this case docosahexaenoic acid. Try spelling that or speaking that when you're slightly tiddly. But this short is DHA. Anyway, you take something that's deficient and you supplement it. In this case, we supplement it with microencapsulated DHA. And then we had a control where we added a calorific control where we added a non-essential fatty acid. And here we can see some of the microcapsules. They're tiny little things, 2 to 1 microns, mixed with some algal cells. And here are the results of the experiment. So we got, this is looking at growth here, we got improved growth with the supplement of 22,6 compared with the non-supplemented algal diet. And there was a slight improvement when we added a non-essential fatty acid. So some of this improvement is due to calorific increase in the diet, and some of it is due to the essential nature of DHA. And then to check that this growth was actually, improved growth was due to the lipids, you looked at the lipid composition of the oysters. And so we found an increase in 22,6 in the oysters that have been fed on this fatty acid supplement, and an increase in triolein in the oysters that have been fed on that supplement. So that was an example of supplementation, and since then many other researchers have followed this pathway and used this approach. So the third leg of the stool of domestication, there's a fourth leg, but I'll tell you about the third leg, is improved desirable traits by selection. And I want to acknowledge the tremendous support we got in the early days from Laverne Weber, who was a past director of the Hatfield Center, and Mark Hatfield, who was probably the third most important person in the United States for a period of time, an Oregon senator. And these two worked together to at least to find funding, earmark funding for MVP for a period of about 15 years. And that got us going, and we started. And this is taking a little time. Here we are. So here's a sort of an outline of our breeding program. We've created 8 generations of selection with 30 different cohorts, each cohort consisting of about 100 families, up to 100 families, not always 100 families. A huge amount of work. And there are many, many people who've been helping with this project over the years. I've just given you here some of the key field MVP managers and geneticists who were very instrumental in making sure that we kept on track. And when we started, we just weren't sure actually whether this was a tremendous waste of public money and effort. And the reason for this was that most domesticated animals are reared under very consistent and constant conditions: temperature, light, and food, abundant food, highly nutritious. And many of them are what's called homeotherms. They're birds or mammals that are able to maintain their body temperature irrespective of environmental variation. Oysters are very different. It's a rough ride, taking a cowboy theme. Tremendous variation in temperature, oxygen concentration, pH, salinity, whether they're submerged or immersed, food availability, quantity and quality, turbidity, all kinds of other particulate material in suspension, and they're bathed basically in a microbial soup. So how do we expect to make any progress when the background environmental conditions that we're selecting for are so variable? And what we decided to do to try and address this question was to select for what we call generalists. So these are families that do well across a wide range of environments. So when we produced a cohort, we tried to plant them at 2 very different sites, either intertidal, subtidal, Puget Sound, or coastal. And so by this means, we hope to basically select for adaptability, if you like. We're looking for families that are highly adaptable to a wide range of environmental conditions. And we were very fortunate. unlike the eastern oyster, which is very picky, and Sam here in the audience can attest to that, the Pacific oyster is very, very adaptable. These are some regression curves here where we looked at the performance of families subtidally and intertidally, and you can see there's a pretty good correlation or regression there, correlation between these two environments. In other words, you know, the ranking of families in one environment is very similar to the ranking of families in another very different environment. And this really surprised and shocked us. Because it meant that we could then select for our generalists. We could go in and find families that perform well in both environments and use them as broodstock to produce the next generation. The other lucky break we had was that we found that the heritability of many of the traits that we were selecting for, yield, which is a composite trait of growth and survival, as well as shell shape and pigmentation, and then in Tamales Bay, resistance to this pathogen, were all pretty heritable, with their heritability values over 0.2, which meant that, in theory anyway, you could select for improvement from one generation to the next. And lo and behold, we could. And hats off to Claudia de Mello here, who did a tremendous amount of number crunching over thousands and thousands of data points to come up with this graph. But this summarizes our progress. So after 8 generations, we were able to increase yields by about 67%. And we did that through not only improving survival, but also growth. So this was very heartening, but it didn't really mean very much unless we could actually get this kind of result in industry, in commercial farms. And we were able to. So we used broodstock from industry that was and produced larvae using industry MVP-derived commercial broodstock, pods at the time we called them. We crossed different pods, we planted the progeny out, and lo and behold, under commercial conditions, we could get a 60% improvement in yield. So this is more than icing on the cake. This is the foundation of the breeding program, to be able to get meaningful improvements in commercial farms in terms of yield and survival and growth. And you can do some rough calculations. If all the farmers on the West Coast produced or utilized MVP broodstock, the total value of the current harvests are about 141 million, times it by 60%. So we're benefiting the industry by $85 million a year. And if you look at all the federal funds that have gone into MVP over 15 years, it's 4.5 million. So we've got a huge return on investment. Problem is not all the oyster farmers are using MVP broodstock. And one or two of the companies have got their own breeding program. Another company is not interested in selection. So there's a lot of variability in utilization of these really, really valuable and meaningful stocks. So one of the things that really changed us and was responsible for a big wake-up call was a discovery in 2018 of a very highly pathogenic variant of this oyster herpes virus. Now, previously in Tamales Bay, that we had discovered, or Carolyn Friedman and others had discovered in 1996, a much less pathogenic variant. And so people were concerned, but not overly concerned. This little fellow here is a different category. And at the time, we thought it's not that it was a microvariant. It turns out that it's not a microvariant, but very similar to a microvariant. And these microvariants have decimated Pacific oyster farming around the world. So literally, a farmer will go to bed one night, and then the next morning he'll wake up, or she'll wake up, and find 95 or 98% of the stock gone, dead. So tremendously impactful disease. And so the appearance of this pathogen, this variant on the West Coast was very, very troubling to the West Coast. And so we did a little pivot in MVP, and we started working on a survival or resistance to this virus. And yield was our second important trait. And in this work, this was really led by Konstantin Dibloff, Tim Green and Ben Sutherland and Spencer Lunder helped in the latter stages where we started to look at the genetic underpinning of resistance. But in these last 4 cohorts of MVP, we really focused on looking at resistance to the herpes virus. And what Constantine found in the survivors of oysters in Tomales Bay, that these oysters had a higher prevalence or abundance of particular markers, or a particular marker on chromosome 8 compared with the oysters that died. And so this marker here was used by Constantine to select for a broodstock to use to produce cohort 30. So this cohort 30 then, it was produced using broodstock that were selected not just based on their phenotype and their pedigree, but also whether or not they had this SNP marker. And as you can see, there was a significant improvement in survival. This is the mean survival breeding value of cohorts 30. And Constantine estimated that we improved our gains by using this marker by about 47% compared with just using pedigree and phenotype selection. So this was a major breakthrough and allowed us to make a much greater improvement in the performance of cohort 30 in Tomales Bay. And subsequently, after a lot of work, we found that a marker on chromosome 8, it was not exactly where this previous marker was, but close to it, was associated with an enzyme called tyrosine protein kinase, SYK. And this particular enzyme is, in mammals anyway, is very important in transduction in the immune system, and it's also activated by viral infections. So it makes sense that this enzyme is somehow tightly linked with this marker. And so in other families, I should say, we haven't necessarily found this linkage to be as tight, and there's some breakdown in that relationship. But that's something that subsequent researchers are going to have to explore. So the other consequence of that 2018 discovery of this highly pathogenic virus in Tomales Bay was that I won't say we, but Gabrielle Serra, who's OSU liaison, congressional liaison person, and Margaret Polaro, who was the director of Pacific Coast Shellfish Growers Association, collaborated and went to Congress and asked Senator Merkley if we could get some help. And Senator Merkley was very receptive and put an appropriation, successful appropriation, to expand the USDA ARS program at the Hatfield Center. So this program is now funded about $2.5 million a year. That's 8 times more than MVP. And it's hopefully going to continue way into the future as long as political interference is avoided. And it includes a program run by Neil Thompson, who is here in the audience, called the Pacific Oyster Genomic Selection Program, or POGS. And this program is going to be very ambitious in that it's using the whole genome of the oysters to select for oysters that are resistant, particularly to this herpes virus. So stay tuned. There'll be very exciting results from that. So I've talked a little bit about the whole process of selecting oysters based on their genetics, on the genetic composition. And I just want to, in the last parts of this talk about a different approach we've been adopting, that is I'm really excited about. And this involves modulation of the innate immune system of an oyster. So an oyster is not like a human. It doesn't have an adaptive, what's called an adaptive immune system, but it has an innate immune system. And it's supposedly not adaptive. But what we found is that we can actually train this immune system. And what has got us really interested in this is the fact that the innate immune genes in oysters greatly expanded. Now, I'm not expecting you to read every label on this complicated graph, But look at the bars. So the height of the bars. These indicate the number of genes in each of these different innate immune pathways. And the yellow striped bars is the Pacific oyster. The purple bars are the purple urchin. The black are the fruit fly, and the pink are the human. And you can see that in a lot of these innate immune pathways, the oyster has a very high number of genes, especially compared with the fruit fly and humans. It's comparable in some of these with purple urchins. So what we decided to do is let's exploit this. Let's see if we can stimulate this innate immune system. And we were very successful. We've been very successful in this. What we developed was a fairly simple process of stimulating this immune system, modulating this immune system. We took fertilized eggs of the oyster and we exposed them to the probiotic mix that we developed. And then after 48 hours, we added a very highly pathogenic bacteria, Vibrio corralliticus. And then after another 48 hours, we checked for mortality and gene expression of the larvae. And here, just visually, you can see larvae with Vicor alone, Vibrio corralliticus alone, pretty much all dead. And then larvae that will be pre-treated with the probiotics and then exposed to the Vicor. And you can see most of them are alive. And then in graphical terms, this is mortality here on this axis. So you can see with Vicor alone, we had more than 85% dead larvae. If you pre-treated those larvae with the probiotics, you could get less than 5% mortality, which is not really significantly different from just the control, which with larvae alone with no probiotics, no corralliticus. So this pre-treatment of the oyster larvae really works like a charm. And Jenny Hesse has done some gene expression work, which really shows how important this pre-treatment is in modulating the innate immune system. So with Vicor alone, we see a very distinct pattern here of genes that are being upregulated. And if you look at their functions in the cell, you can see some of these are related to, associated with this NF-kappa B pathway. This NF-kappa B pathway is very important in oysters in inflammatory responses and responses to diseases. So this pathway is being activated by at least these two genes. If you treat the animals, pre-treat the animals with Vicor, with the probiotics, a very different pattern. And if you look at the cell and the genes that have been turned on in the cell, they're very different from the genes that have been turned on by Vicor alone. So we're hypothesizing then that this modulation of gene expression in some ways prepares the larvae for subsequent exposure to this highly pathogenic bacteria. And this obviously is an enormous amount of work that needs to be carried out to continue this. So in summary, I'm kind of wrapping up now a bit. We haven't defined the artificial diet. We don't have a commercial artificial diet. This is a big problem for hatcheries and farmers in general because we don't have a cheap alternative to algae. And for the biochemists and researchers, we don't have nice tools to try and understand the nutrition and biochemical pathways of oysters. So this is a big challenge for the subsequent generation of researchers. So I'm very enthusiastic about this continued selection to improve farm yields and disease resistance. We've got solid funding with ARS program and a really good foundation to continue this work. We've really only just started to scratch the surface on interactions between oysters and microbes. So the oysters are a big blob of protoplasm, if you like, that are sitting in a bacterial soup with about 1 million bacterial cells per mil. In their guts, they've got swarms of bacteria that are helping them digest and process food. How do they prevent those bacteria, many of them are pathogenic, from invading their tissues? Well, they have a suite of methods to do that, mucus and cilia and things like that. But we also need to get a better understanding of of the oyster's innate immunity. How does the tremendous number of genes that oysters have compared with humans and fruit fly, how are they activated? How do they respond to these different bacterial threats? And then, of course, viral threats are another issue. So with the onset of the appearance of this highly pathogenic virus, can we vaccinate, if you like, in inverted commas, the oysters against this virus so that they're not going to be not going to suffer the high mortalities that have been seen around the world? So my oysters are in a corral here, but they're still jumping around. So there's a good chance that they're going to jump out. Anyway, I want to thank so many people who've helped us over the years. The funding sources, shout out to USDA, NIFA, and ARS programs, the NOAA National Sea Grant Aquaculture Program and SK programs. All of these are essential. And with all the funding cuts ahead of us or proposed by the current administration, all of this is in jeopardy. And then, of course, the Pacific States Marine Fisheries Commission that has funded all this recent work we've done on looking for SNP markers for survival of oysters in response to herpes virus. A shout out to the industry partners. So again, none of this, particularly the MVP program, would not have been possible without close collaboration with a whole group of industry partners: Whiskey Creek Hatchery and staff, Alan Barton, Sue and Mark Weigert, Goose Point Oysters and Dave Nisbet, Oregon Oyster Farms and Lou Chin, Bill Dewey and Taylor Shellfish Farms, Hog Island, Terry Sawyer, John Finger, Gary Fleener. All of these people have been really, really important. And hats off to Margaret Polaro and PCSGA for going to Congress and getting funding for us for the establishment and expansion of the ARS program. And then for technicians, really, these folks are the people who are the wheels under the car. They keep us moving forward, and without them, we just simply would not, as researchers, be able to meet our objectives. So in terms of MVP, Blaine Schoolfield, Jeremy Jennings, Dave Jacobson, Anya Robinson, these people really stand out, Kirill as well. And then with the probiotic work, Candace Thorstensson has really taken the lead in making sure that this work progresses and also is extended now to commercial hatcheries. So we're working with Whiskey Creek on applying some of those probiotics. And of course, many people who I've not mentioned here, wonderful friends and colleagues, and there are just too many to mention here. But MVP data analysis, Claudia de Mello has really been very important in the last 5 to 8 years of MVP in helping us analyze data and apply modern quantitative genetic techniques to select for broodstock. Tim Green, Ben Sutherland have worked very closely with Constantine and myself in genetic analysis. And then way back in the distant past, Roger Newell, Danielle Krieger have been instrumental in helping us understand the nutrition of oysters in a natural environment. And then passing the oyster to the next generation of tamers. Many, probably over 30 graduate students over the years. Just some shout outs here. Daniel Krieger, I mentioned, Matt Gray for oyster feeding physiology, Matt Hawkyard for looking at microencapsulation nutrition of marine suspension feeders, fish larvae as well as bivalves. And then with oyster breeding and genetics, Constantine Divloff and Evan Derland, Spencer Lunda, Sean Matson, all been a part of that story and produced wonderful dissertations and theses. And then on the probiotic story, Jenny Hesse, I've mentioned, and David Madison. These two master's students did wonderful work on isolating probiotic bacteria and finding out how they affected the oyster larvae. So I guess we're on time. Isn't AI wonderful? Anyway, that's my sunset picture. Thank you very much.
Thank you so much, Chris. I appreciate that. We're going to bounce back and forth between online questions and those in the room. So, Roseanne, any questions online?
Yes, I have one from Bill Han Shoemaker. He says, "Hi, Chris. Aragonite and seawater pH. Could you explain their relationship?"
Oh, yes, okay. So pH is a measure of the acidity of seawater. And when people talk about ocean acidification, they really often refer to pH, how acidic is the water. Aragonite saturation state is a little different, but it's related to pH because very often as pH drops, as the water gets more acidified, so the solubility of aragonite, which is a form of calcium carbonate, and the main isoform of calcium carbonate in the shell of larvae, that aragonite becomes more soluble as the pH drops. So as it becomes more acidic, the aragonite becomes more soluble, which is a problem for larvae, because larvae, especially in their first 24 hours or so, are depositing aragonite like crazy to create their shells. About 70% of the dry weight of an oyster larvae, 24-hour oyster larvae, is made up of aragonite. So in these, they're like race cars. These organisms have to produce this shell very quickly. Any perturbation in that causes problems for their development. Interestingly enough, the Australurida, which I mentioned right at the beginning of my talk, is kind of a lazy Ford, and it gradually produces its shell over a period of several days. And that species is actually much more resistant to acidification than the Pacific oyster. So maybe we should look at the Ford again, not the Maserati. Yeah.
Thank you, Chris. How about questions in the room? Of course. Hang on just a second.
Exercise for you, Cinnamon.
Folks online, I'm running up the stairs. You're hearing me breathing because I haven't done this in a little while.
Hey, Chris.
Hi.
Obviously an amazing amount of work done over a 4-decade career, lots of accomplishments, really moving it forward. But as you laid out, lots of challenges that lie ahead. So what is your prognosis for the Oregon oyster industry moving forward?
Well, it's really, in many ways, a canary in the coal mine. I mean, not just in terms of the susceptibility of the larvae to ocean acidification, but also they're very susceptible to water quality, changes in water quality. And so with coastal development that's occurring increasingly, more and more people are moving to the coast to retire, like myself, and recreate. There's more pressure on water quality. And so farmers are increasingly struggling with that and trying to ensure that the quality of their product, their food product, remains exceptionally high. So I think the industry has some challenges, but I think with goodwill from the public, I think the word is called what society's license or something like that now, and with political support, then I think then the industry can flourish. And there's a tremendous potential for oyster farming to benefit the environment. So tremendous amount of work has been done, particularly in Delaware and Chesapeake Bays, on the importance of oysters in creating habitat for a lot of benthic organisms, for clarifying the water, cleaning the water of suspended material, and generally stabilizing the substrate and preventing erosion. So these are some of the benefits of oyster farming. And many of the NGOs now, like World Wildlife Fund and the Nature Conservancy, are waking up to that fact, and the public as well, as that oyster farming actually can be very beneficial to the environment.
All right, go ahead, Roseanne. I think a question online.
How are oyster industries in Australia and New Zealand recovering from OSHV1?
Oh, good question. Yeah, so first of all, they got some government help because initially there was devastation of the industry and a lot of small farmers were facing bankruptcy. And then the breeding programs got into place. So luckily this resistance to this virus is fairly high. The heritability values are fairly high. And so you can develop resistance in 5 or 6 generations. And so the Australians, the Europeans, and the New Zealand folks have developed breeding programs that have bred for greater resistance. They still see mortalities, but they kind of compensate for those mortalities by simply planting more seed. Now, we are a huge advantage here on the West Coast because we're anticipating, we're ahead of the game. We've anticipated this as being a potential problem for our industry. And so we've established already a breeding program with ARS to look at this specifically and to hopefully develop some very resistant strains before this virus escapes from San Diego, assuming that it does.
Thank you. How about questions in the room?
Chris, thank you for a really wonderful presentation. I was just wondering, for the Japanese oyster, is the genetic diversity here on the West Coast a subset of what is in Japan? And have you considered going to Japan and perhaps maybe some of those are better suited, especially for what our habitat is becoming? I'm sure you've talked to Japanese researchers, but just wondering if that's something you've thought about.
Well, that's music to my ears, Laurie. So we've already done that. In 2004, myself and a couple of colleagues went to Japan, and we went to southern Japan. So most of the oysters that are farmed on the West Coast originated from Miyagi, which is in northern Japan, where winters are really cold. I mean, they get ice on the ocean and snow and everything else. So in 2006 or 2004, we went to Japan, not actually to find some alternative to the Pacific oysters. We're actually looking for Kumamoto oysters, but we ended up collecting these Pacific oysters because at the time we really didn't know what we were doing. We brought these back, and these were from the Ariake Sea in Kyushu Island. And this sea has water temperatures in the summer of somewhere in the region of 28 to 30 degrees centigrade. And they have herpes virus as an endemic disease in that bay. So the idea was that when we brought these animals to the US, we went through a huge amount of quarantine work to make sure that we weren't bringing any diseases with them, that we would have an alternative broodstock to the Miyagi strain that was more temperature tolerant and more resistant to the herpes virus. And at the moment, this stock is of great interest to the industry because it has a very deep cup. So it's a little like the Kumamoto. I think it's a major species of the ARS breeding program, and they're evaluating its resistance to the herpes virus and resistance to temperature, high temperatures in the summer. So I think the jury is still out on that sort of on those other questions. But some companies have embraced the species because of its nice shape, deep cup.
Thank you, Chris. Okay, Roseanne, go ahead. Question online.
In all your travels, what's the best oyster you've had?
Oh, well, I think when you eat an oyster, you're not just eating the oyster, you're participating in the whole environment in which you're sitting. And I think that there's one example where I really had a wonderful experience, which was in Edinburgh, where I did my undergraduate degree. There's an oyster bar called the Royal Bar, and it's all Edwardian inside, so it hasn't been changed. And Edinburgh wasn't bombed during the Second World War, so many of the buildings are intact. And so sitting in this Edwardian bar, eating European oysters at that time was one of my most memorable occasions. Yeah.
Nice. How about other questions in the room? Roseanne, any other questions online? Okay, we got another question in the room. Hang on, everybody. I'll get there.
I know you've worked a lot on artificial diets. What do you think the future holds for that, especially for larvae?
Oh, yeah. Well, I think the lipid supplements are something that will be helpful because larvae basically accumulate lipid as they develop and use that lipid during metamorphosis, because during metamorphosis, they can't feed. And so that's a major energy supply. So I think, and that technique is already utilized, and we already know how to make lipid emulsions and lipid supplements. I think the other approach of trying to replace algae completely with an artificial diet is still in the distant future. I was very arrogant when I was a young professor here. When Annie Robinson was around, I looked at her algal vats and I said, "Oh, we're going to replace all those," you know. I daren't look at her now because I know I made a huge mistake saying that. But there's some real challenges because basically you have to create a living cell. And to do that, you have to have a lot of control over these different parameters I mentioned in my talk. So it's a technological challenge. But I think liposomes actually have a lot of potential in terms of developing, of retaining the water-soluble nutrients, and then combining them with these alginate beads or cross-linked protein particles. Whether they be economically viable, that's another question. But I think as a research tool, I think I can see that on the horizon as being possible.
All right, we're going to end with one last question online. Go ahead, Roseanne.
Thank you, Chris, for everything you've done to support the shellfish industry. One of the hallmarks of your career is the way in which you've engaged the industry. As we've seen in recent months, the connection between publicly funded research and a benefiting industry or community is extremely important. What advice do you have for students in finding connections between their work and real-world applications?
Well, I think my advice to students is to listen to the farmers and the hatchery operators. Go and talk to them because they see things way ahead of us as researchers. We're not in our ivory towers, but we're close to that. And so the OA problem, for example, I really wasn't aware that this was a serious problem for commercial hatcheries. I thought it was something just going on in our local Hatfield hatchery when we started having problems rearing larvae. It wasn't until we talked to Sue and then we talked to the other hatcheries on the West Coast, we realized this is a major problem. So I really encourage you to engage with industry and the farmers. They're at water level. They see what's going on a daily basis. They see long-term trends. And they often point out really important research questions that have not only applied value to their farm, but also raises some really interesting fundamental research questions. For example, you know, the probiotic work that we're doing. We're really looking into the immune system of oysters and how that immune system functions. And I think understanding that is going to have huge repercussions, not just for oysters and oyster farming, but also for human medicine. So, you know, that's oftentimes the research ideas really are germinated by contact and correspondence interactions with industry.
So, Chris, we're going to wrap up there because we're at time, but we've got some more questions online.
Okay.
Can people still reach out to you and ask
Yeah.
questions?
Yeah, sure. Yes, if folks have questions, I'd like to email me or give me a call. I'd be happy to answer them. Yeah.
Perfect. We are standing in the way of a party. So if you are not aware, there's a retirement party being set up right outside the door. So if you have follow-up questions, you can reach out to Chris at that. For folks online, if you want to reach out via email, that would be great. But for everybody in the room, let's give Chris a round of applause.
Oh, thank you. I feel you.
Thank you, everyone. It's
Thank
been
you,
a real
everyone.
pleasure working here at the Hatfield Center with you all. And I'm not going away completely, so it's going to be a gradual slide into oblivion. Thank you once again.
Thank you very much, Chris. We'll see everybody either outside or in 2 weeks. Thanks, everyone.
Hi.
Way to go.
Yeah.
I'll do a little announcement.
Okay, great.
15, 20 minutes.
Okay, good.
Okay, so
Yeah.
get out there, get some food.
Okay, all right.
Okay.
Great, thank you. Was that okay?
Still learning stuff. I mean,
Oh, good. All right. Well, I didn't know whether it was one of
the history stuff. I was like, oh, this person was really. Like, I didn't know a couple of those names. What was your advisor's name again?
Oh, Peter Gabbett, Peter Warren.
Okay.
Yeah.
Okay.
So he was a giant in his time.
Yeah.
He passed away when he was only 53.
I saw that. Yeah.
Yeah, very early. He got cancer, liver cancer, hepatitis. I think he ate too many oysters. That's my guess. Mm.
Not funny, but yeah.
But anyway.
You're very welcome. All right, for folks online, we're going to end the presentation. Thanks so much for being here.